Well, I'd like to thank the organisers. I'll try and get us back on track. Maybe turn this into a consulting exercise, because what I notice from my position, I'm the head of consulting for a clinical research organisation, and often what the academics are doing and what the investors want tend to butt heads, and if I can impart some wisdom and help you get funded to do what you're doing with these marvellous vaccines, that's a good day for me. So if I was going back to the academic bench and I started a BSc at the University of Queensland and Professor Hall was teaching back then, how would I do things differently? Well, this is a slide we see from lots of vaccine companies, and they talk about the billions of dollars in the 12 years, and these estimates come out of Tufts University economic assessments. The actual direct cost of human vaccine development is around about 50 to 150 million. We're running a vaccine study at the moment where the sponsors put in 100 million to their program to date, and we'll probably spend about 80 million on their phase 3 program. That's a therapeutic vaccine. What you have here in blue is the regulatory clinical development stages, and this will be applicable to an animal or a clinical vaccine, and then in yellow your manufacturing stages, and then in purple the sales responses. And at the very early stage, I think it's important to do elements of all three, because this is what the investors really want to see, is have you considered these points so that you can get from where you are with proof of concept in my studies to actually getting your vaccine into a human or a cattle or an animal that's going to prove the potential of the vaccine in the target market where you're trying to sell your vaccine or take it further as a spin-off company out of a university. And all these things are important. We've heard a couple of discussion points around manufacturing today, and from the NIH presenter, for example. About 3 months ago, I'll give you a real-life example. We had a scale synthesis method for an anti-infective that involved hydrazine. Now, hydrazine is what we use in rocket fuel. It's also genotoxic. So, although you can do it at small-scale synthesis Sorry, I'm just being motioned up the back there.
Just don't tap the lectern.
Oh, I see, sorry. So although we can do it at small scale, when you actually get it to commercial scale, it needs to be safe, scalable and economic. And this is why these early considerations for how you're going to manufacture your product is very valuable early on. So we concluded in our report to that particular client that at best case, at the scale they wanted, which was 285 kilo of this anti-infective, best case scenario was a laboratory fire or a factory fire. Worst case scenario was an explosion, given the quantity of hydrazine they actually needed to use to make this particular compound. So I like to really visualise these things as a web of events, and there's pockets of expertise that you need to draw upon, and you need to develop partnerships and relationships. And there's the basic research that you're all familiar with in this room, candidate selection, manufacturing, toxicology, the patent position of your program, the sales and marketing, socialisation with the WHO, for example, your product vision, your key concepts, and getting all this pulled together and financed. And I think at the centre of this is a development plan. We advocate them heavily from our perspective because it sort of ties in all these contingencies, and it puts a cost and a timeline around what you need to do to reach that first milestone and get it invested. So what do investors want to see? Well, they want to know that the vaccine can be sold. It's the primary concern they have. Now, that can be for an emerging market at very low cost of goods sold, for $1 per vaccine. So for vaccines that we discussed around rotavirus. But somewhere we've got to make sure that there can be a profit made somewhere that can be reinvested back into that program for public health benefit, or someone can make a commercial gain out of it, because there needs to be a return of investment. And we had a client recently present at a conference where there's high net worth individuals who want to invest in socially responsible programs that see global benefit, but they need to see a return on their investment, and that was the Cavendish Foundation in the US. So tailoring what you're doing with your vaccine to some of these economic principles is valuable up front. So I'm just going to give you an example of a program that we took to license a few years ago. Being a CRO, I find it difficult to present data from clients because it's confidential. We're keeping it confidential for a reason. But this is published and it's also registered, and this vaccine is now owned by Sanofi Pasteur. So the early work was done very well and proved a lot of key points, and it was done at the University of St. Louis, and again the NIH presented on that point before. So they were able to show in small animal models that the vaccine was immunogenic. They were able to show in a primate, they were able to manufacture the vaccine in a Vero cell line, which is an established cell line for human vaccine manufacture. And it wasn't virulent, neurovirulent, because this particular virus, Japanese encephalitis, is neurovirulent. They were able to show that the attenuated live virus wasn't neurovirulent when intracerebrally challenged into mice. And they were able to characterise those specific attenuation sites that resulted in that attenuation. And it's things that the regulators want to see. They want to understand mechanism of action generally, and being able to show attenuation and risk of reversion in that data is very helpful up front, because they'll always raise that, particularly the live attenuated vaccines. So this is the step where it was then in-licensed into a small biotech company called Orovax at the time, and later became a Canvas. And they were very good at publishing and keeping investor interest in their program through good publications. If you want to look at how to socialise investor-related science, the Canvas model is a good one, because they had journal editors knocking at their door all the time just to submit their publications. It was a novel technology, and they were able to, get some momentum behind their science, which was good, and it paid off for their investors, and I'll show you that slide later. So this team was able to show that in a very stringent model in the macaque, that if you vaccinated the animals and you challenged them intracerebrally into the brain, that the animals that were challenged were vaccinated, survived the challenge, and those that were not, did not. And obviously what they were able to demonstrate is that the immune response developed quick enough that the tiny fraction of antibody passing the blood-brain barrier was able to protect against an invasive infection. That was quite artificial, but proved the point. So we had proof of concept in a very relevant model. Not that Japanese encephalitis infects that way, but it was able to demonstrate in a severe way the potential utility of this vaccine. So from these studies, they were able to put some key visions around this vaccine and how it would work, because there was a number of problems with the predecessor vaccines, and this is what we call with the jargon, the target product profile. So what are we trying to achieve with this vaccine, and how are we going to, what's the vision for the vaccine? Well, really we wanted a one-dose vaccine, and we wanted it cheaper to make so that we could compete with the existing 3-dose vaccine that was made in mouse brains and was formalin inactivated and was quite reactogenic. So that was the vision behind this vaccine. Make a better vaccine that's easy to use and is better tolerated. And we knew that the protection was very low with the commercial vaccines, so we wanted something that would actually provide protection over at least a 5-year period. So what we did is we wrote down a series of key claims, and then we went about proving these key claims through our clinical development plan and our other activities. So did we know the mechanisms of action? Could we justify that to the FDA? And we felt that that was predominantly antibody related. Could we compete with a 3-dose schedule compared to a 1-dose schedule? Did we have a correlate of protection? Now, at the time we didn't, but the WHO then published halfway through the program that a correlate of protection would be a titer of 10, and that was based off Charlie Hoke's studies in Thailand many years ago with 50,000 paediatrics. So we did get a correlate of protection that was widely acknowledged, and it made our program much simpler. And we also had to demonstrate that we could protect against numerous wild-type strains. There's at least 4 genotypes in JE, and it was safe both in adults and paediatrics, because the target market in Asia is the paediatric population. That's who you're trying to protect. So price, we had to make sure that, if we were going to compete with 1 dose, with a 3-dose schedule, how much could we charge for this 1 dose? And So there was some analysis around this. We knew that the 3-dose schedule was $200, and we knew that the competing vaccines coming in from the CSL were $120 per dose. So we had a price range of at least $200 to $240 for a 1-dose vaccine, and we could undercut these vaccines by either going lower than the 200 mark or charging a premium for convenience, so that your traveller could come in a Western market where you're going to make your returns, because you're trying to subsidise what you're trying to do in key markets such as Asia to prevent this vaccine-preventable disease in children. We could actually target the traveller market and say, oh, 1-dose vaccine is more convenient, you just need 1 dose and you can go off on holidays. So the price point, I think, came in at around $250 and $280 a dose. And of course, we were competing with Vero-derived vaccines that were inactivated, so we had to make sure our cost of goods sold would allow these price margins with a return on the investor. So with our formulations, we actually started with a wet frozen formulation. That's quite common and that's quite acceptable. The regulators understand that. So it was a basic formulation. And then when we knew we had proof of concept, we actually did a bridging study later on, and we lyophilized and we confirmed that the immunogenicity of the 2 formulations was the same. And of course what we did is we're very careful to use excipients that are widely acknowledged as safe and are widely used in other vaccines. So there was nothing too sporting in our formulation there that would cause concern to the regulators. So the manufacturing program was really around an intensive Vero culture, and we were able to increase the scale of manufacture by annealing Vero onto the microcarrier beads. This was a Baxter invention at the time, and it led to far higher yields, and we were able to control that yield at scale. And of course the other things we were conscious of is the adventitious agent risk and controlling that, and also reducing risk of hypersensitivity by controlling preservatives and also antibiotics. The standard specifications are listed there. So the upstream process was really culture in Vero, and the downstream process was a series of purification and filtration steps and making sure that we were removing any advantageous risks. So nothing really particularly difficult there from a manufacturing scale. Building simplicity into vaccines is a good idea up front. It goes back to that point about looking at your scale and looking how you can do things, because you don't want to make it complex for a commercial manufacturer. Drug development gets complex all by itself. So our animal studies really follow the key ICH guidance. If you look up ICH M3R2 and ICH S6, there's an alignment across the world on what studies should be done. Really, you have to prove that, through challenger immunogenicity, that your vaccine works, and then you've got to do a series of experiments that shows it's not toxic. The safety pharmacology, obviously this is a neuroinvasive vaccine, and JE can actually cause disseminated organ damage, visceral organ damage. So we were looking at key organ toxicity, and particularly the CNS. So that's what we did in our safety pharmacology. Generally, for vaccines, you don't have to do genotoxic work unless there's something in your program that produces a genotoxic impurity. And our toxicology really mirrored what we were doing in terms of the vaccination in the clinic. Something I'll add here is with these live vaccines, we wanted to know where they were going, where they were replicating in the body, and we wanted to know a genetic stability of the vaccine. Was it changing once it was growing inside the host? So there are some elements that will be specific to your vaccine, and I will put more emphasis now on embryo-fetal toxicity. FDA, TGA placing more emphasis on that now when 10 years ago when this vaccine was done, they didn't have the same emphasis. And it's a realisation that vaccines will be used in pregnant women, and it presents a risk that needs to be addressed in your toxicology program. So that's the current expectation. So in our clinical program, we looked at roughly 3,500 subjects. Now, we were able to reduce that number because we had the correlative protection that was recognised by WHO. But then we had to repeat a lot of that work, the dose ranging studies, the safety efficacy studies in children and toddlers, to prove that we could use this vaccine in both adults and paediatrics. So the total total study numbers are about 11 all up. Now generally the WHO for a prophylactic vaccine asks for 5,000 subjects, so we were under that because of our correlate allowed us to come under that, and we did have pre-studies that supported using that correlate. So these are the sort of studies that are actually conducted, and a lot of these are published as well, so you can go and have a look at how this vaccine was developed. If you go to the websites like the TGA and the EMEA and the FDA, you'll actually find what they call European Public Assessment Reports or summary basis of approvals, and they give you a good idea of how vaccines have been registered in the past. And it really gives you an insight if you've got a very similar vaccine or you're trying to do something that's similar to a licensed vaccine. It gives you a clear path on what you can borrow from, as well as obviously the public literature. But what the regulators will want to see is, have you got the right dose? Is that dose safe and is it effective? Using a recognised field study, so vaccine efficacy, or a correlate of protection that's widely acknowledged and justified. You can see that the dose ranging and the memory response, long-term immunogenicity and the vaccine interaction studies were all done at phase 1, phase 2, and then we did a pivotal phase 3 program followed by a confirmatory phase 3. It was in the confirmatory phase 3 that we actually did our lot-to-lot assessments and confirmed that our 3 commercial scale batches of vaccine were actually going to give the same geometric mean titers. That's something the FDA likes to see. And then once we had got our license for our adult population, we started into Philippines, Latin America and Asia to be able to develop the toddler and the children program, and that was actually taken on by Sanofi at the time. So we used 2 key regulators to develop this vaccine. One was FDA, obviously. The advantages of FDA is they don't charge you for advice, and TGA doesn't either. They actually sting you at the end, so your filing fee at the end for FDA is roughly 2.2 million currently. This is where they get all the free advice costed back. But at early phases of development, you can open an IND, have a pre-IND meeting and get good advice from these key regulators internationally. Because this vaccine was actually a GMO, we had additional considerations with the Office of the Gene Technology Regulator. So we did have discussions with that group as well. So how did the whole program go? Well, actually we came under the average. It took us 11 years from start to finish. And from where it started on the bench, with the good work from the team from St. Louis University, to where it was signed by a canvas, which was Oravax, and then the marketing work that was done and the eventual sale of Sanofi canvas to Sanofi, which was £285 million. And now the vaccine's registered in India, Thailand and Australia, and they haven't done much with it for a reason I don't quite understand, but I've lost touch with the program. So what would I do again if I was in the position of an academic trying to license a vaccine? Well, again, I would want to answer some core questions up front that would support discussions with investors. What's my vaccine vision, indication and key claims? How can it be sold and reimbursed in key markets? You don't have to do every market in the world, but you need to consider your key markets. And US is usually a good place to start. Where is my vaccine now? Where do I want to exit? Do I want to spin this into a company and develop and sell the company, or do I want a platform? Give some considerations, because that affects your IP position, and in Australia certainly your Australian R&D tax reimbursement, which is important. It's half price, 2 for the price of 1 here for those companies that are eligible for that tax reimbursement. And then I would really write a plan that addresses the key aspects of manufacturer, regulatory and clinical development, and also your sale, your cost recovery, and how it's going to work. And those plans are usually 20 to 40 pages with an executive summary, and then you can make a decision tree out of that. And from that, you can distill that really into a slide deck what we call the pitch deck for your investors. You're going to need to talk to key people, regulators, investors, potential buyers under confidentiality, and be mindful of your IP position and your publication strategy prior to closing your IP position, and have some professional help if you don't have that inherent in your team to actually talk with the regulators as well as investors. It's not a coffee chat with regulators. They expect the discussions to be data driven, and investors want clear things when you meet with them, so you don't want to waste those opportunities. It's going to be a busy schedule if you're an academic and you're teaching and you're doing running a research program. So, but the worst case scenario is to pull someone with key knowledge out of a company because then it will flounder. So you've got to think about strategies for keeping invested in this small company and bringing in the management expertise and using contractors and keeping your team virtual and cost effective while you're committed to these other programs and other activities in your professional life. And that has to be managed. And again, what I've highlighted here in yellow is some of those core competencies that you'll need to consider. Project management, your partnerships, your communication, investment, finding investment that's both dilutive and non-dilutive. And when I say non-dilutive, that's grants, and dilutive is where the VCs take a big chunk of your company. And then the necessary expertise to sort of put all this into a plan and a framework that's commercially time-driven. If you're going to license this vaccine after your patents have expired, you're not going to get the investment in your program. It has to be in a commercially sensible time frame. So I'd just like to thank a number of people that worked on the Campus program with me, and my current colleagues who have contributed to this presentation. And some declarations there. I've consulted for a number of companies, and this is general investment advice. Thank you very much.
Okay, thanks Mark. Are there questions?
I was interested in the comment about the vaccines affecting the fetus. Were any studies done on pregnant females?
No, that's a good question. So the yellow fever vaccine has been given to a number of women who have been pregnant, and there's been no adverse effects. But in general, you'd contraindicate a live attenuated vaccine during pregnancy. The issue arises at what happens if someone is vaccinated and they don't know they're pregnant, and how do you predict the risk there? So that's done typically in two species, and we call them the segment 1, the segment 2 studies. And New Zealand White Rabbit's one of them, and we can use a rodent species for another. And what we're looking for is effect on the embryo. We're looking for birth weights of the pups. We're looking for all these things that could indicate that if this vaccine does go into a pregnant woman, there's going to be a problem. And it's a standard battery of tests. Even though there's a contraindication, the regulators get nervous when they see programs that haven't addressed this battery of tests prior to licence. And the timing is variable. Certainly for women of childbearing potential, when you're doing your early phase clinical studies, using highly effective contraceptive methods and really informing them of the risk is acceptable to most ethics committees. But as you start to expose more and more women, you're going to have to do those studies because of the risk that something will slip through, and you would certainly have to do them prior to phase 3.
There's obviously an explosion of nanoparticle delivery technologies. Are any of the regulators worried about the long-term effects and build-up of those in recipients of vaccines?
I've just had a nanotechnology meeting with the FDA last year, and it was actually a plastic nanoparticle. And we felt that they'll be very concerned about that because there's no way to actually remove that nanoparticle from someone. And they were not concerned about it. And we were asking whether they'll treat it as a device or a vaccine, and they confirmed that it will be treated as a vaccine. But what they wanted to see is where it was going, the biodistribution, any genotoxic impurities that were associated with the manufacture of that plastic. And there's a tagging issue because I think one of the other presenters earlier said, with the sand, you can't actually see these particles. You can only indirectly see what they're doing because of the histology. So we actually tagged the particle and we traced it, we looked where it was going, and it was sitting there for quite a while and it wasn't going to clear. But the issue, the FDA came back and said, well, immunologically, will that tagged particle behave differently than your untagged particle? So I think that they're abreast of the issues, and but as long as you can answer key questions, you know, because a lot of this, I won't reveal the plastic because of confidentiality, but we inhale and ingest tons of this stuff in our lifetime. So having, you know, nanoparticles in the skin, the FDA just agreed that it wasn't a core concern at that time. So, interesting.
Any other questions? Okay, if you'd join me in thanking Mark.
Thank you.